Eh bien, bonsoir à tout le monde dans ce webinaire de la NSFA sur le cholestérol trafficking and atherosclerosis. I think I'm going to have to switch to English because we have speakers that come from the United States. Good. We are very lucky today to have three outstanding speakers. Dr. Laurent Calvier, can you hear me? Everybody's, is that okay? Can you see me? Can you hear me?
I can hear you and see you.
Okay, very good. So the first speaker will be today Dr. Laurent Calvier from UT Southwestern Medical Center at Dallas. He's going to talk about monocyte recruitments and the role of renin, anti-renin against atherosclerotic lesion formation. We have also another outstanding speakers, Professor T.Y. Chang from Dartmouth Medical School in Hanover, United States. He's going to talk about ACAT1. T.Y. is the one who is actually identify and characterize deeply ACAT1, this important enzyme, into the ER. And we have another outstanding speakers, Dr. Fabien Alpi. He's research director at the IGBMC here in Strasbourg. And he's going to talk about contact sites, how cholesterol can traffic, not through vesicular trafficking, but directly by contact site between organelles. So just to follow up on this, can you see my screen? Yes. So cholesterol trafficking is, as you know, very important for cholesterol homeostasis, and it's usually knows and was described a long time ago from Mike Brown and Joe Goldstein that cholesterol enter the endocytic pathway through LDL receptor or through cholesterol-rich domains, and cholesterol esters into the lysosome or the endocytic pathway needs to be hydrolyzed by the lysosomal acid lipase, and this releases the free cholesterol. Then the free cholesterol needs to egress the lysosome. For this, there is cholesterol transporter, namely NPC1 and NPC2, and I think TY is going to talk about this in detail. Free cholesterol reaches the ER or other membrane inside the cells through vesicular transport or contact sites. And Fabien will talk about this contact site, especially contact site between the ER and lipid droplets. And free cholesterol reaches the ER where it's hydrolyzed, where it's esterified, with the ACAT1. So this important enzyme that re-esterifies the cholesterol to downregulate cholesterol biosynthesis or eventually accumulates it through lipid droplets. So Laurent Calvier especially works on anti-renin therapies that prevent monocyte adhesion and foam cell formation, and as you can see here, protect against atherosclerotic lesion formation in mice, but also apparently in human. It's also a powerful anti-inflammatory drug therapy. T.Y. Chang isolates, purified, and characterized deeply ACAT1, and he especially developed inhibitors of ACAT1 that protects against atherosclerotic lesion formation. There is a lot of translational actually interest in that webinar today. And we also have Fabien Alpi, which talk about this contact site system where the cholesterol can actually traffic from an organelle to another, not through vesicular transport, but directly. And for this, it uses different type of proteins that transport the cholesterol. So let me introduce you first to Laurent. Laurent, can you share your screen?
Sure. Let me do this right now. Okay. Okay, do you see my screen? I think so, right?
Yeah, it's working. Very good.
Okay, should I go ahead?
Yes, please go ahead. Yes.
Okay, great. Thank you very much for the kind invitation, Philippe. So as you mentioned, I'm going to present my work on Relin, the over APOE2 ligand, which promotes monocyte recruitment in atherosclerosis. So briefly, oops, sorry, I have a problem here. Okay, now I'm back. So atherosclerosis is characterized by the deposition of fats, cholesterol, and other substances in the inner arterial wall. And this buildup or plaque causes the arteries to narrow, blocking the blood flow. Eventually, they can also burst, leading to a blood clot associated with life-threatening stroke risk. The good news is that atherosclerosis can be treated, like healthy style habits can help to prevent its prevention, like healthy food, for example, sport, or when you decrease a risk factor like smoking or drinking. In addition, a wide range of cholesterol-lowering medications has been developed, such as the statins, resins, cholesterol absorption inhibitors, the fibrates, the niacin, and lately the PCSK9 inhibitors. So to control the concentration of cholesterol in the blood, there is one receptor family key for cholesterol trafficking, the low-density lipoprotein receptor family. So this is the family here, and as Philippe mentioned in the introduction, about like 50 years ago, the pioneer work from the Nobel's professors Braun and Goldstein, who are actually in my department, led the identification and characterization of the first member, so the LDLR, here on the top, on the left, and they identified this receptor during the search for the molecular basis of familial hypercholesterolemia. The LDLR has been extensively studied for its preeminent role in cholesterol homeostasis and the removal of LDL from the circulation. Thus, for this historical reason, the biology function initially attributed to all members of this gene family was a role in lipid metabolism. However, since the initial discovery, many of the functions have been unraveled for this family. And for this presentation, I will focus on one receptor, the APOE2 for apolipoprotein E receptor 2, also known as LAP8. So as suggested by its name, this receptor binds ApoE, which combines with other apoproteins, cholesterol, and other lipids to form the lipoproteins as illustrated here. There is different isoform for ApoE that differ by single amino acid substitutions, which affect their functions, but I'm not going too much into those details today. So briefly, in the vasculature, ApoE binds to ApoE2 to stimulate endothelial NOS production and endothelial cell migration. ApoE2 also plays an important role for endocytosis. It recognizes extracellular ligands, including ApoE, and internalizes them for recycling or degradation in lysosomes via a process of receptor-mediated endocytosis. And surprisingly, and unlike suggested by its name, ApoE2 is hardly known to play a role in lipid regulation. But it's expressed also in neuron, and where ApoE2 is essential for neuronal migration and positioning in the developing brain. In the adult brain, this receptor modulates synaptic plasticity, migration of neuroblasts, as well as dendrite and dendritic spine formation. And outside of the central nervous system, global deletion of ApoE2 in mice has no effect on plasma lipoprotein profile. So that suggests a minimal or redundant contribution in plasma lipid regulation. So going back to the brain, ApoE2 has another well-known and major ligand, that's Relin. So this extracellular protein was initially recognized only for its role in guiding neurons during brain development and as a synaptic homeostatic regulator. However, renin is also abundant in the blood, but its role there was completely ignored for several decades. Therefore, by looking at this second ligand, we opt to demise a mechanism to stop atherosclerosis progression independently from cholesterol-lowering strategies. So this is really intriguing, right? Because if I summarize, we have on one hand an LDLR family member, ApoE2, expressed in the vasculature, but this receptor appears to have a limited effect on lipid concentration. And on the other hand, we have renin, a well-known neuroguidance protein, which appears also to circulate in a significant amount in the blood, but its function there is unknown. And like I mentioned, in the brain, this renin ApoE2 so ligand receptor, they are both beneficial. So we hypothesize that they have also a protective function in the vasculature. So to study this question, we have at our disposal a great tool, monoclonal mouse antibody, CR50, targeting and depleting ring from the plasma, as you can see here from these plots. Okay, so first we tried atherosclerosis models. We used LDLR knockout mice with a high cholesterol diet, and we observed in both renin conditional knockout, so on the top here, and in CR50 treated mice in the bottom here. We observed a reduction of plaque area, as you can appreciate from these representative photos here and their quantification. So basically, we expected that renin depletion would exacerbate atherosclerosis, right? But we observed actually quite the opposite results. So that was really surprising. So basically, it has a protective role in the brain, but looks like it's actually the opposite in the circulation. All right, let's move to the next one. Here we go. So to understand this phenotype, we looked at the infiltration of leukocytes in the aorta, especially monocytes, and they were clearly reduced in renin conditional knockouts as suggested by the MAC3 staining here. Similarly, the expression of endothelial adhesion markers, VCAM1 and ICAM1, were also decreased in renin knockout mice, which makes perfect sense, right? Because if you have less adhesion proteins on the endothelium, you also have less adhesion and infiltration of leukocytes into the subendothelial space. So this observation would suggest that renin promotes monocyte endothelial cell adhesion. And we looked at the adhesion of monocytes by intravitreal microscopy in the marginal mesenteric vessels, because those vessels are easier to image because they have a really thin vessel wall. And we used CX3CR1 GFP mouse line, which are genetically modified to express GFP in monocytes. So that makes it really easy to track. So let me guide you here through the image and the video that you will see. You should recognize the autofluorescence wall here on the side, and in the lumen, you see the green monocytes that will follow the blood circulation. So I'm gonna show you now the movies. They might take a few seconds to load. Let's see. Okay, I think here we go. So this is what you see during the recordings. On the left, we have a control mice treated with control IgG, and on the right, treated with CR50, so the anti-renin antibody. And here, try to focus on the rolling monocytes, not the fast one flowing with the circulation, but the slow one. And you should be able to notice two effects. So CR50 decreases the number of rolling monocytes, and also they roll faster. So we quantified those videos here. So this is the analysis. You see here that CR50 decreases the number of rolling monocytes and also increases the velocity. So consequently, when you stack all the images from the movie for 10 seconds, so this is the cumulative picture here, you clearly see a decreased exposure of the endothelium to monocytes. And you have here the quantification for the cumulative intensity, and the rolling index is actually the count divided by the velocity, both showing you that there is a decreased exposure of the endothelium to monocytes. Okay, next one. So here, so next we, to explain why CR50 reduces monocyte rolling, we looked at the expression of adhesion proteins. And as you can see here, that was done in the aorta. CR50 markedly decreases the expression of rolling adhesion molecules, so E-selectin, and the firm adhesion molecule ICAM1. So this was performed in mice, and we next wanted to know what about the humans, right? Of course, we couldn't do it directly, so we used human cell cultures for this purpose. And first, we tested human monocyte adhesion to human aortic endothelial cells in vitro. So endothelial cells were treated with plasma, so human plasma, combined with or without CR50 antibody. So this is what the conditions you have here. And in the quantification here, you can appreciate how CR50 blocks monocyte adhesion either at 1 or 24 hours. And as a side note, we also obtained similar results with direct ring stimulation instead of using human plasma. So we also investigated the cellular pathway in human endothelial cells, and so as expected, ring induces the expression of VCAM1 and ICAM1 and E-selectin. And it's interestingly, when this induction, we also showed that this induction is mediated by ApoE2, the renin receptor in those cells. As you can see here, ApoE2 knockdown blocks the induction of adhesion markers. Following ApoE2 binding, we have also shown that renin induces a cascade of phosphorylation through PI3 kinase, AKT here, A-kappa B alpha, and finally NF-kappa B. So to summarize this first part, we have shown that renin binds to ApoE2 here, activate the PI3 kinase, AKT, and NF-kappa B pathway, which induces the expression of adhesion proteins such as E-selectin, and we showed ICAM1 and VCAM1 as well. So thus, renin increases the adhesion of leukocytes to the endothelium, allowing their infiltration into the subendothelial space. So going back to our initial question, we asked, can we prevent atherosclerosis without progression independently from cholesterol lowering strategies? Well, here we've demonstrated that the LDLR family member ApoE2 and its ligand renin represent an interesting target to control the endothelial function directly, and consequently control also the inflammation. So this renin ApoE2 mechanism, which control the endothelial activity, is also important for many other diseases. Let's see if the next one is coming. Such as multiple sclerosis, arthritis, and I'm sure that just like me, you want to know if this vascular mechanism is also reproducible in different inflammatory contexts. So although this is slightly different from atherosclerosis and our topic today, I would like to give you a sneak peek on some other inflammatory driven diseases that we've been studying in the lab. So the first one is multiple sclerosis, or MS. So we induced autoimmune encephalomyelitis, or EAE, in mice treated with IgG as a control, or CR50. So EAE is induced by immunization with myelin, reproducing the human MS. As expected here, you see in the plasma that CR50 depleted renin expression, and really interestingly, also all the treated mice survived, unlike the control group. We have also evaluated daily the paralysis using a scale from 0, where there's no clinical sign of paralysis, to 10. So 5, half of the body is paralyzed, and 10, the mouse is dead. And as you can judge here on the first graph, CR50 delays the onset and also moderates the severity of the paralysis. This is also reflected by the weight loss, the middle graph here, and the hanging time. So for the hanging time, we put the mice upside down on a grid for up to 3 minutes, and it reflects both paresis and paralysis. So here you can see that CR50 protects from paralysis, and to be closer to clinic aspects where you rarely treat in prevention, we have further tested CR50 as a treatment. So starting on the first day of paralysis. So as expected here, renin was depleted in the CR50 treated mice, but surprisingly, if you look at the control group, renin is increased by EAE induction. So that would suggest that there is a specific response to inflammation here. Also importantly, all the mice survived, and you can appreciate that the paralysis was, the progression of the paralysis was blocked by CR50 treatment. And if you look at the 5 last days, they actually start even to recover here. So based on this unexpected increase of renin expression in EAE mice, we looked at plasma concentration, renin plasma concentration in patients with multiple sclerosis. And we found that renin is increased during the relapse here, and was back to control level during the remission phase. So renin concentration in human also correlates with multiple sclerosis severity. So the next thing we did is to look at the mechanism, and if CR50 protects from monocytes extravasation during EAE. So the mice we've used for our EAE experiments express GFP under the CX3CR1 promoter, meaning that they will express GFP in monocytes, which is great for us, but also in the microglia. So to be able to distinguish infiltrating monocytes from the microglia, we have stained the microglia with a specific marker, IBA1, here in red. So consequently, infiltrating monocytes appear in green only, as pointed out by the white arrowheads here in the merged picture. So in addition, there is also fewer inflammatory cells overall, as you can judge by the total amount of GFP positive cells. So we've quantified this histology, and this is what you see here, the decreased infiltration of monocytes. And importantly, this decrease was observed at all EAE stages here. And this is how we understand these results. Renin, by binding to its receptor ApoE2, here promotes the expression of adhesion marker via NF-kappa-B, and consequently, renin promotes the adhesion and the infiltration of leukocytes or monocytes into the CNS, so into the spinal cord and the brain. And this propagates the inflammation and the demyelination. So you understand that blocking renin stops this cascade event and protects from paralysis progression. Finally, I'm going to finish my talk by presenting some unpublished results obtained in arthritis. So this is a very promising result that we have here. We've measured renin levels in serum from patients with rheumatoid arthritis, and you can clearly see here that renin level increases in patients with arthritis. And this was correlated with arthritis severity as judged by the disease activity score and the sedimentation rate. So based on these observations in human, we moved to inflammatory arthritis mouse models. So first in the KBXN serum transfer model, which induces acute inflammation. So mice were treated in prevention with CR50. And you can appreciate here that both the clinical score and the PAO swelling are reduced when renin is depleted. And to confirm our results, we used the collagen-induced arthritis as a second model, which mimics better the initiation phase of the autoimmune reaction, collagen type 2. Here you can see that CR50 treatment, starting at the onset of the symptoms, prevents the progression of the disease as judged by, again, both the clinical score and PAO swelling. So these two inflammatory arthritis models suggest that renin depletion by CR50 prevent the progression of the inflammation. And so to conclude my talk, I will emphasize one more time on the mechanism that we have unraveled and how the organism reuses a neuroguidance protein, namely renin, to sort of pave the way for leukocyte extravasation. So we have demonstrated that renin binds to ApoE2 on the endothelial cells to activate NF-kappa-B pathway. And as you may know, NF-kappa-B pathway is well known to tune the status of this endothelium by regulating the expression of many pro-inflammatory genes in the endothelial cells. So target genes include ruling molecules such as E-selectin and firm adhesion molecules such as ICAM1 and DCAM1. Therefore, the endothelial adhesion followed by endothelial transmigration here are both regulated by NF-kappa-B target genes. However, you don't want to directly target or block NF-kappa-B because it's a major signaling pathway for cell function, and most likely a direct obliteration will result in adverse side effects. So instead here, we suggest to modulate its overactivation during inflammation by targeting upstream regulatory pathways like renin. And during chronic autoinflammatory or autoimmune diseases, this pathway is activated and the endothelium becomes more adhesive and permeable. So our lab has shown that renin depletion restores the endothelial function and prevents inflammation in atherosclerosis, but also in MS or arthritis. And with this last slide, I would like to highlight and thank all the collaborators behind this work in the different labs, Hertz Lab, Karp, Stuve, and Scholz. And thank you all for your attention, and I will be happy to take any question you might have.
Okay, Laurent, that's really beautiful, beautiful data. Congratulations. We have time right now for maybe one or two questions. Then we can come back on if you have additional questions at the end of the webinar. But if we have time now for one or two questions from the audience, if there is any. I may actually have one. You test for athero, you tested in LDL receptor deficient mice. Have you tested that also in other athero models like LRP? Because the LRP deficients, they obviously have larger lesions.
Yeah. So I haven't done it on LRP1, for example, but we're working on a different model that's directly on ApoE2. There is some mutations that promote the activation of this receptor. So we're actually working on those models right now.
Okay. Other questions from the audience? Okay, so thank you very much, Laurent. You stay around, because we may have additional question at the end of the webinar. It's now for me a great pleasure to welcome here at the webinar Professor T.Y. Chong from Dartmouth, University of Dartmouth, United States. So please, T.Y. Can you share your screen? Good.
Let's see.
Hello, sir.
Yeah, some technical problem.
Share screen.
Share the screen. Share.
Yeah, share the screen. Good.
Is that okay?
Yeah.
You see it? Okay, great.
Yeah.
The first slide, I think. Okay. Yeah, thank you. Okay. Well, sorry, I always have some technical difficulty. So I'm going to talk about this enzyme AK1 SO1 blockade to treat cardiovascular neurodegenerative disease. And this is, as you can see, it says T.Y. and Kathy Chen. Kathy is the long-term collaborator. We work together at Dartmouth and Geisel School for more than 40, almost 50 years. And we like to watch birds. This is, in case you don't know, this is an indigo bunting. Kathy has a quick hand, so she take a picture. I just fly into the cherry blossom tree. Next one. Okay. To people who don't know about us, we work on the enzyme called AK-acyl-CoA cholesterol transferase 1. And in case you don't know anything about AK1, we'll just give a brief introduction about biochemistry, cell biology, and eventually medical physiology. And okay, this is cholesterol. It's essential for growth of mammalian cells. It's a lipid cell membrane. And cholesterol is a precursor for steroid hormone, bile acids, oxysterols, and transport cholesterol, vascular or non-vascular transport. Okay, for those audience who don't know anything about how it exists in the Chen lab, the Chen lab at Dartmouth, we actually started with living with a Chinese hamster ovary cell, so we make mutants. You can see from here, the A is wild type cells. The CHO cell is a very good model, and you can isolate mutants. And the mutants, when isolated, actually is useful to isolate the new genes that come by gene complementation. For example, this M1, M19 is a mutant we isolated a long time ago. And the mutant sent to Mike and Joe's lab, they identified the gene S2P gene. As you know, S2P is an SRIBP processing pathways needed at the Golgi, cook the SRIB, liberate the SRIBP. And the second mutant is called 25RA. And this mutant, you can see, this kind of like an experimental foam cell, a lot of lipid droplets. And that's because this mutant has a mutation, and the mutation, it has a gain of function. It just continues to produce cholesterol. Turn out that cell was used again by Gossi and Brown's lab. They identified a gene called a SCAP. That's a cholesterol sensor in the ER, as you know. Well, maybe we'll talk about the fourth mutant. The fourth mutant is a CT60. This is isolated by Ken Cadigan, graduate student in the lab. And this mutant is very interesting. You can see this is by differential interference contrast, a lot of particles that's accumulated inside. And it's different from lipid droplet from the one on the left. That's the cholesterol ester lipid droplet. This stuff on the right, the cholesterol rich. It's cholesterol filled with endolysosome. It turned out this is a CHO cell model for the it's a CHO cell model for the MPC mutant. And that mutant was used by Peter Pincher at NIH. He used that mutant to identify a gene. That's the MPC gene. Okay, I will talk a little bit about MPC later. And then finally, the last one, that was actually say that's the bread and butter. This thing you can see, it's very clean. It doesn't have a lipid drop like the one in the left. It doesn't have the particle on the right. It's very clean. And to identify the ACAT gene, that's a gene that makes cholesterol ester from cholesterol. You can see this mutant has no lipid droplet, nothing. And we've been living on this ACAT for a long time. So as I said, that CHO cell was a good experimental cell, sort of like a foam cell. Well, here's the real foam cell, and this is a macrophage filled with cholesterol esters. You can see that's the business end of atherosclerotic plaque, lots of cholesterol ester and lipid droplets. And this is done by Richard Anderson. Now, as I said, our labs have been living on this enzyme called ACAT. And sometimes the name is called SOT. It's sterile O-acyl transferase. The turn out the ACAT is in the gem bank. In the gem bank business, the ACAT is actually a different enzyme. It's not our enzyme. It's not the SOT. Our enzyme in gem bank is actually called SOT. So we call now ACAT1 SOT1. Okay, so that's cholesterol, oleo-CoA, and eventually for this is cholesterol ester. And this enzyme is first discovered Alvin Slater. And then the gene is the cholesterol storage in Hashimoto and Brown-Goldstein, I know. So the enzyme stay as ACAT. So we never really changed the name. We just call it ACAT1 SOT1. This is really in honor of the people who discovered the enzyme. And the gene is cloned in our lab. Some of the not so yet elderly audience may remember the ACAT1 gene knockout mouse was done in 1996. Okay, when the gene is cloned, but this spot freeze. The gene knockout, I will tell you more about the gene knockout experiment. Okay, there's some controversy about what exactly does ACAT1 do to atherosclerosis. Okay, and there's some other misconception. I'd like to actually clarify based on this opportunity. As I said, we purify the enzyme, and eventually this enzyme now is actually on the map. The structure has been solved by 3 groups. Yan Yin's group, this is Chen Long's group, this is Li Xiaochun Li in Dallas. Okay, and the paper is published 2 years ago. And then there's the ACAT2. Okay, I'd like to clarify what exactly is ACAT2. Okay, there's misconception about ACAT2. The ACAT2 eventually first was identified Robert Young, Steve Sturley in yeast first, and then mammalian cells, Larry Rideau, Bob Faris, Steve Sturley. Okay, so here's just we did the biochemical grinding work. Okay. Here we show the enzyme is activate cholesterol oxidative allosteric regulation, and regulated, and it doesn't really change the protein. Okay, you may wonder how does ACAT regulate? It's regulated by cholesterol oxidative allosteric. It's not regular SRBP. It's not the regulation not at gene level. It's homotetramer, and the active site it's in the 7th transmembrane at the tip of the transmembrane. We can show that the enzyme can esterified pregnenolone. Right, this paper really set the pace for the allosteric control, and we show that enzyme is actually a 2,4 dimer. Eventually, the structural biology is solved. We're very lucky. We collaborate with Chen Lei in Beijing University. As I said, the structure is solved by 3 different groups. Okay, here's the picture. So 7 carbon, it's got 7 transmembrane, and it's got the histidine tag. It's pretty much support the biochemical attack, and it has the blue color it shows binding site ACAT. Now this is important information because the binding site of ACAT seems like in the hydrophobic, very open range. This is very interesting information to me. It says that ACAT inhibitor, why are there so many ACAT inhibitors? Well, it turned out because the hydrophobic, it is a hydrophobic pocket. And yeah, the blue stuff is hydrophobic pocket. And this is the histidine. Okay, so it blocked the histidine. And that's very useful information because basically that's how the ACAT inhibitor works. Hydrophobic molecule get in the pocket and block the histidine accessibility. This is transmembrane 7. Okay. Now we think the ACAT is regulated by allosterically by oxysterol pregnenolone. Now how does that work? We think there are two binding sites. There's a substrate site, and there's also a substrate site activated site. When this is activated, somehow activated, and that's an allosteric regulation. I mean, this is just a model, and this allosteric regulatory site actually will take a long time to work it out. Okay, now this is show how does ACAT get involved with atherosclerosis. This is actually a very important work was done by Tom Bolkane. He has an ACAT inhibitor, CI976. Okay, so this is the rabbit, cholesterol fat rabbit, and there's a lot of foam cells. Okay, this is the macrophage stain. Abundance macrophage, lots of macrophages. And this is interesting. When you feed the ACAT inhibitor a low dose, they don't change the serum cholesterol concentration, but the macrophage become much less. The foam cell drop. It's really eye-opening. So that leads the idea that ACAT actually can prevent the atherosclerosis at the local level, not the serum cholesterol level. Now, unfortunately, the clinical trial showed the ACAT inhibitor, the CI1011 developed by Pfizer, and there's another one, Pacnamide, developed by Sankyo. They failed to act as supplement to statin to further reduce serum cholesterol, so it got dropped. And this is the reason the ACAT inhibitor was dropped, because they do not work as supplement as a statin, and not because the ACAT inhibitor was toxic or doesn't work. Okay, these are misinformation in the literature. Okay, and I would like to clarify the misinformation. Well, this slide shows the ACAT. The ACAT is actually over the body, and it's in the brain, and it's in the steroid hormone producing cell macrophage, and there's some in the chylomicron. The ACAT2 play a major role in the cholesterol absorption. Okay, basically in other tissues, basically it's all ACAT1. Okay, there's a misconception. Says ACAT1 may play a very big, ACAT2 play important in liver, VODL cholesterol. That is good for monkey and good for mouse. It does not work in the human. Okay, in the human, the VODL is cholesterol, ester, poor, and triacylglycerol rich. Either whether it's ACAT1 or ACAT does not play a major role in the liver. It's just for human. And if you apply the data in a monkey and a mouse to human, then that's where we get into trouble. Okay, so the ACAT2, if you want to block ACAT1 or ACAT2 and try to reduce cholesterol VODL, well, it works in the animal model. It does not work in the human, because the human in the liver, the ACAT1 level is very low. ACAT2 level is also very low. Now, so exactly what? So how can? So the question. The question is why still continue to work on ACAT? Well, it turned out there's some interesting result, which I will show you. The brain is cholesterol rich. Cholesterol play an important role in the brain. A small portion of cholesterol is stored in the cholesterol ester. The cholesterol ester mostly in the brain is mostly by AK1. And that cholesterol ester cannot substitute the function of cholesterol. That's a key. So what does cholesterol ester do? People say, hey, that's a lipid droplet. Okay. To form the cholesterol ester is inefficient utilization of cholesterol. If cholesterol ester accumulate, it's inefficient accumulation for cholesterol ester. It's inefficient utilization of cholesterol. That's to us. Okay. So we want to block the ACAT and create a more efficient utilization cholesterol. Okay. That's so I don't know whether it's a new concept. Our data suggests that's what it is, and as I will show you in the Niemann-Pick type C model, that's what we'll show you. Okay, now this is in the brain, in the triple transgenic mouse. As you know, some of you may have followed the Alzheimer's disease research. Alzheimer's disease, there's a triple transgenic. So there's APP, there's oligomeric beta, and there's also tau. So with this genetic knockout, the AK1 blockade, that's from the gene knockout of Balfour's were breeded with a triple transgenic mouse. And it was actually beneficial, okay? And this is important because it suggests that blocking ACAT in the brain, it will reduce the A beta rescue cognitive deficit. It's not, at least it's not toxic. Okay, there's another misconception. So blocking ACAT is toxic. That is misconception. Okay, it actually restored the cognitive deficit. We cannot see any difference. It did not hurt the brain. And mechanistically, how can it work? We showed that in the ACAT1, if you block increased autophagosome, these are published. I won't bother you with the data. Okay. It's published more than 7 years ago. It's increased autophagic flux, stimulate lysosomal degradation. Okay, so basically somehow when you block the ACAT, the cholesterol ester can actually restore the function of endolysosome. Okay, it's I guess it's strange. Okay, if you think that when you block ACAT, increase cholesterol, it should actually fill should actually fill the lysosomal cholesterol, or you should have formed the cholesterol crystal. That is not what we see. In the brain, if you knock out ACAT, the cholesterol decreases. There's no cholesterol crystal. Okay, we reproduced it. That kind of phenomenon in the atherosclerotic model. So what's the mechanism? Now, it not only in the triple transgenic mouse model, it not only reduces APP, A beta, it also reduces tau. Okay, so autophagy. Okay. Now, so this is more just an observation. So as I said, the ACAT1 knockout, and there are folks that think that it's actually damage will actually not benefit atherosclerosis. It's controversial. The AK1 knockout in atherosclerosis, the data from Balfour's and from Ishibashi, they do not agree with each other. It's controversial. And that's just 2000. In 2007, the AK1 inhibitor become available. The AK1 inhibitor is done by paroi. They feed the atherosclerosis. It reduces atherosclerosis. Now, our lab did a genetic lab. We make a myeloid AK1 knockout. Myeloid, okay, macrophage, neutrophil. Okay, if you do a lysom-free breed with AK1 knockout, our data support Ishibashi's work. And it when you block ACAT, it actually benefits atherosclerosis. We also show that this is when you use myeloid AK1 knockout, it suppresses diet-induced obesity. As you know, diet-induced obesity increases adipose tissue macrophage. We showed if you block the ACAT in macrophage, it actually reduces. These are the papers. Okay, we published 3 papers. Now this is myeloid ACAT. Just reduce ACAT in the myeloid macrophage. It will. What happened? It reduces lesion macrophage. It slows down macrophage migration. So there's actually in the atherosclerosis, there's less macrophage. It's not what the literature tell you. It says macrophage die in the atherosclerosis. That is actually wrong, because when you block the ACAT in the macrophage, their migration slow down. And there's actually not enough monocyte-macrophage in the atherosclerosis. And there's not, and the interpretation, some labs says that because macrophages die. Well, actually, that's because macrophage never went there, all right? And we published that a few years ago. And we did the same thing, myeloid-specific ACAT1 ablation, attenuating the inflammatory response to macrophage. In other words, in the dye-induced obesity, there's macrophage adipose tissue, and then we examined in the macrophage, in the myeloid-specific ACAT1 knockout, there's actually less macrophage. So it supported the work in the atherosclerosis, but there's simply less macrophage in it. And we follow up. Some predicted, it says if you knock out ACAT, cholesterol will actually increase, and they will actually fold in the endolysosome, form cholesterol crystal. Well, we use advanced lesion model. If you do for the mouse for about 20 weeks of high cholesterol, high fat diet, and the cholesterol builds a high cholesterol. In the myeloid ACAT1 knockout, there's less cholesterol. Okay, so it's the other way around. It's not as literature predicted that there's less macrophage, and because of the diet, cholesterol crystal, that goes without data. If you do the data, there's less macrophage. It doesn't enter, and there's less cholesterol. There's less cholesterol crystal. The ACAT blockade does not increase cholesterol crystal. That's in the literature. It's a wrong literature. Okay, so we need a hypothesis to explain what's going on. How can you block the ACAT when you increase cholesterol? It actually benefit. Okay, here's the hypothesis. Well, we think that because cholesterol binds phospholipids, sphingolipids, form lipid raft. It's lipid raft, and cholesterol is needed lipid raft. And that lipid raft, the cholesterol binds to the phospholipid. The phospholipid is called a mobile cholesterol. This is not our hypothesis. Ivan Lange test stack. This is the kind of stimulus. And the mobile cholesterol can move from one membrane to the other, to the Golgi endosome plasma. In other words, if you block ACAT, that cholesterol will move. Why? Because cholesterol in excess of binding, they can move. Where do they move? They move to Golgi endosome plasma membrane. So here's the hypothesis. So when you block ACAT1, expect to increase the mobile cholesterol at the ACAT1 domain. And the mobile cholesterol will move away from the domain, increase the cholesterol content in other membranes. So in the Alzheimer's disease, they disrupt lipid raft. So when you disrupt the lipid raft, you block ACAT1, you will repair the lipid raft, because that mobile cholesterol move to provide more cholesterol to the, I believe the same model can apply to atherosclerosis. All right, in other words, in atherosclerosis, that's cholesterol accumulation. That's inefficient utilization of cholesterol estrogen. Now we use the model, mouse model, in MPC disease to test A1B hypothesis, because in MPC, here's MPC disease. Well, that's a rare genetic disorder, mutation called build-up cholesterol. Okay, no, remember this. In MPC, the cholesterol build up in the endolysosome or in the spleen, liver, brain. Sequestration of a lipid in endolysosome, starvation Okay, this is the follow-up. When the cholesterol is sequestered in the lysosome, they're sequestered. And that means the cholesterol in multiple other membrane organelles, there's a result in neurodegeneration, okay? And that's a fatal, so it's MPC1, MPC2. So we use MPC to test the hypothesis. So if block AK1, because MPC collect in the lysosome, the cholesterol cannot be utilized very, very well. If you block AK1, it'll prevent the cholesterol be utilized, and that should help MPC. Okay, here's the model. In the AK1, the lipid droplet. Now the lipid droplet is formed by cholesterol that move by endosome dependent or some other process, okay, through contact sites or non-vesicular transfer, and form the ACAL. ACAL form the cholesterol after. And when you block the ACAL, that cholesterol will be reutilized. The mobile, this is the mobile cholesterol. Mobile cholesterol they move to the plasma membrane, and they move to the alveoli, and they will be utilized by ABC1 and for cholesterol disposal. So in our argument, when you block AK1, it releases the cholesterol. It should actually it should help utilize the cholesterol, and then should get rid of cholesterol. Okay, this we believe the AK1 blockade activate the cholesterol disposal pathway. And this is the test AK1 knockout MPC. Well, indeed, the lifespan, the MPC mouse, they only live about something like 100 days. And when we just do the AK1, this is total AK1 blood. It's not a myeloid, it's a total AK1 blood. This is about apheresis model. And the lifespan increased by 34%. Okay. Well, I think 34% is not a bad deal. I'm an old guy, and if my lifespan increased by 34%, I will be very happy. Well, this increased the weight. MPC mouse before they died, the first in the animals before they died lose the weight. Okay, and in the double knockout, the weight increase. The weight loss is protected, and so I won't bore you with the data. To give you a summary, it increases lifespan, slow down the body weight, and it delayed Purkinje neuron loss. So it affects the neuron. And for atherosclerosis, one want to know, at least it's not toxic in the brain. Okay, here's some data that show the syntax is 6. That syntax is 6 is a cholesterol required. The proper localization syntax is a T-SNAP. It's involved vesicular trafficking in the TGN, trans-Golgi network. It's proper localization. It requires cholesterol. Okay. Now, the syntax in protein, AK1 knockout does not change the protein. But the syntax in 6 localization is completely different. The syntax in 6 in the MPC knockout, it becomes mislocalized. You can see it's not localized at the one edge of the nucleus. Syntax in 6 is when it's properly lipidated, the TGN is at one side of the nucleus. And you can use a reflecting angle, you can measure it. And the MPC cells, it's mislocalized. You lose the TGN localization. Syntax 6 is not properly done because this is the reflecting angle. And there's another Golgi. Golgi is a membrane tether in the trans-Golgi network. And how about the Golgi 97? Is that also? Yes, the syntax 6 is mislocalized. Syntax 6 and the Golgi 97, they're both mislocalized. In the double knockout, they both combine. Okay. And the quantitation, this is the quantitation. Okay, and the quantity they come back, mislocalization, double knockout, bring it back. And this is another control. GM130 is a cis Golgi, and it's not affected much. So the NPC mutation affects mostly TGN, and GM130 is not. It doesn't change. So we did have some control. GM130 doesn't change. Okay. So one would say you must be, well, if in fact the syntax is sex, TGN, endolysosyl, then the cation-dependent manner 6-phosphoprotein, manner 6-phosphoreceptor receptor, that should actually be altered. Yes. In the NPC mutant, the manner 6-phosphate receptor is very, very much down. This is shown by Susan Pfeffer and Yakum. This has been shown before. Okay, so double knockout bring it back. Cas7D is a major protease in the endolysosyl, and the process in the Cas7D is screwed up in MPC. Now, if the endolysosyl recover, then the Cas7D should get recovered. Yes, it did, because the monocytes is recovered and Cas7D is recovered. So there's a conclusion. Mutant cells, NPC cell, the membrane failed to receive cholesterol and late endosome, they reside. So the syntax said Golgi, they are aware that means to localize, inhibited, come back. And it also restored the protein content of manner 6-phosphate receptor Cas7D and ABCA1. AK is a new target for the NPC cells. Okay, now the last couple of minutes, I want to clarify some of the misconception about AK inhibitors. Now, if you read literature, there's a lot of AK inhibitors. I mean, it's very boring. Okay. One of the, I should clarify, ATR101. This is, you can see the highest AK inhibitor, wow, very high K, very low Ki. Inhibit AK1, doesn't touch AK2. And this is failed in preclinical trial. Okay. So this ATR101, it turned out it's an extremely toxic AK inhibitor. Okay. And so literature says, don't use AK because this is very toxic, and the ATR101. And thank God there's a lab in University of Michigan that re-examined the ATR101, indeed inhibit AK1. It also inhibit ABCA1. It also inhibit ABCA1, ABCG1 gene expression. No wonder it is very toxic. It has a huge off-target side effect. So if you use AK inhibitor, please do not use ATR1. It's a disaster. Okay, and this is why the AK inhibitor brings so much controversy. Okay, now how about other inhibitor? Yes, there are at least 3. CR1011, CR1011, K604, and F1125, F. How do we get it higher? Can't see it. Oh yeah. F12. I want to emphasize this F1251 is discovered by Pierre Fabre in French. Okay, and this is indeed a very high AK1 inhibitor. It's not toxic. It's got abandoned because it does not complement a statin. Okay, K604. Again, this past clinical trial, these last 3. Oh, okay. Oh, sorry. Yeah. We don't have to go drill the structure. Okay. Please do not use. There are at least 3 AK inhibitors that actually passed clinical phase trial 1. Now we want to focus on the F12511. We like it because it's pretty potent, inhibited mostly AK1, not so much on AK2. It's hydrophobic, and it's difficult to prepare surfactant. And Pierre Fabre used either trin-80 or cyclodextrin. Okay. Now we worry this may be actually causing trouble. You take it, calamine pathway, and enrich it adrenal toxin. So we develop a nanoparticle. This contain DSP, EPEG, and phosphatidylcholine. We will put hydrophobic. Okay, so this, and we inject in the blood. This DSP, EPEG protect the particle from getting degraded. So the half-life of the particle stay in the blood and stay much longer. And the size is about 110. This is published stuff. And we tested. We can show the F12511 in 1 hour, in 4 hours, able, which is measure enzyme activity. We can show that in 4 hours, inhibit AK in the brain. It also inhibits the adrenal gland. In the 8 hours, it still works. The K compound only works in the adrenal gland. It does not work on the cerebellum. K compound cannot.
TY, I think we are running a little late, so okay. I don't know how many slides you still have.
We'll finish. Yeah.
Okay.
Well, this is it's not toxic, and we did the kinetics of it. And the half-life of this compound in the blood is about 4 hours. And this is the last slide. And this is Kathy on the left, and the middle girl is adrenal. She played a key role in developing the F compound. Yeah. So this is my last slide. Yeah. Sorry about it. Yeah.
So it's no big deal. Okay. Thank you very much, TY. I may have some questions, but we are running late.
Yeah.
Maybe in the audience, there's also some questions. So maybe if you stick around, we'll talk about all this at the end of the webinar after Fabien's talk. Fabien, are you there?
Yeah.
So Fabien Alpi is a research director at IGBMC, and he's going to talk about cholesterol trafficking through contact site.
Okay, do you see my screen?
No, we're still seeing the TY screen. TY, can you stop sharing? Fabien, can you try to can you try to share your screen?
It should be shared. No, it does not work.
No.
I'll try again.
TY, you still there? Okay. Okay. All right. Perfect.
Does it work?
Yeah.
Yeah. Okay. Okay, so yes, so I will talk to you about cholesterol transport at interorganite contacts. And okay. Just to work, okay. So as we already heard in this webinar, atherosclerosis is characterized by the formation of lesions in the wall of arteries. And one main characteristic of these lesions is the formation of foam cells, which are these cells that accumulate neutral lipids, and more specifically, sterol esters. And these foam cells can derive from macrophages that accumulate in the lesions, or also be derived from vascular smooth. And while it has been shown, as previously mentioned, that the level of circulating cholesterol is key for the formation of foam cells, and that led to the development of several therapeutics that are used in human. We don't know exactly how cholesterol accumulates in foam cells, as the pathway that cholesterol follows to reach its final destination in lipid droplets is not totally understood for concerning the intracellular part of its trafficking. So what do we know about the distribution of cholesterol in cells? So what is known is that cholesterol is unevenly distributed in cells. So this is a scheme that shows cholesterol concentration that is color-coded here with this green code. And cholesterol is mostly found in the plasma membrane, as shown here. And it also accumulates, but with a lower concentration in the endocytic pathway, and in another form, not in the form of free cholesterol, but in the form of esterified cholesterol, it can accumulate in lipid droplets. So what is a bit surprising with this repartition of cholesterol is that actually cholesterol can be synthesized in the endoplasmic reticulum, which is one of the cellular compartments that is the poorer in cholesterol concentration. So actually there are two sources of cholesterol for cells, either biosynthesis, which occurs in the endoplasmic reticulum, or an exogenous source, such as LDLs or lipoproteins. These lipoproteins are recognized by specific receptors, as we heard from the first talk, and with the complex of the receptor coupled with lipoprotein is endocytosed, and it reaches late endosome lysosomes, from which cholesterol is desesterified and can be transported to several areas of the cell. So just to, I just want to mention here that I will come back on this endocytosis event, and I will talk to you about another receptor for later, that is transferrin receptor. And transferrin receptor takes a similar route as LDL receptor. So it is a membrane receptor, and it enters the endocytic pathway, and it can be recycled back to the plasma membrane. So what is important for atherosclerosis, as TY mentioned, is that actually this kind of cell accumulate cholesterol, sterol esters in lipid droplets, and it is important to understand how cholesterol, either from the endogenous pathway or from the exogenous pathway, reaches its final destination where it accumulates in foam cells. So what is known in eukaryotic cells is that there are two main mechanisms that allow the transport of lipids in cellular compartments. So as you all know, lipids are hydrophobic, so they are not easy to be transported by their own. And so they are transported either by vesicular transport or by non-vesicular transport. And this non-vesicular transport involves specific class of proteins that are named lipid transfer proteins. And I would come back on the description of these proteins later. So the first question that can be asked is cholesterol mostly transported by vesicular transport in cells or by non-vesicular mechanisms? And so well, almost 20 and even 30 years ago, several labs addressed this question, and I will just show you the work from Fred Maxfield's lab that they published in 2002 in this JBC paper. And in this paper, they used fluorescent analog of cholesterol that is named dehydroergosterol, DHE. And this DHE can be added onto cells, and it first reaches the plasma membrane. So this is what is shown here on this panel A. So as it is fluorescent, it can be easily followed in cells. And when it is added onto cells, it first reaches the plasma membrane, and when cells are incubated with DHE, you can see that it progressively accumulates intracellularly in this dot that has been identified by Fred Maxfield lab as the endocytic recycling compartment. So what the authors of this paper have asked is this transport of DHE dependent on energy, meaning that it would most likely be mediated by vesicular transport, or is it independent of energy? And so to test these two possibilities, they depleted energy in cells by treating them with these two compounds that deplete ATP. And they looked first as a positive control at transferrin receptor. So as I told you earlier, transferrin receptor is a transmembrane receptor that is endocytosed when it encounters its ligand. And this is what is shown here. So transferrin receptor is labeled here at time 0. It's found on the plasma membrane. And when you allow the receptor to encounter its ligand, it is endocytosed and it is found in recycling endosomes. And in cells that are depleted for energy, it cannot be endocytosed. So meaning that transferrin receptor internalization requires energy, which is what is expected for vesicular transport that mediates the attachment of vesicles onto microtubules and that require energy to be transported. And then they perform the same experiment, but this time using DHE. So at time 0, DHE is found in the plasma membrane. In normal condition, it reaches the endocytic compartment, as shown on with this dot that accumulate at the perinuclear region. And in cells that are depleted for energy, the DHE transport was not blocked at all. So meaning that sterol internalization is energy independent. So this and other work suggested that cholestérol, the intracellular transport of cholestérol, is mostly mediated by non-vesicular pathways. So how are these non-vesicular pathways working? So these non-vesicular pathways involve lipid transfer proteins, and in human there are many different protein families that consist of lipid transfer proteins, and this is shown here on this scheme where you have a list of all the lipid, well, not all, but most lipid transfer proteins that have been identified. And some of these lipid transfer proteins are known to be involved in the transfer of cholesterol, such as OR proteins, start proteins, and start-like proteins. So how do these proteins work? So as I told you, these proteins work, most of them, they have an hydrophobic pocket in one of their domains, the domain that binds lipids. And this hydrophobic pocket allows them to bind one molecule of lipid and then to transfer this molecule of lipid to an acceptor membrane. And the lipid domain, the lipid binding domain, acts as a shield to protect the lipid molecule from the hydrophilic environment. And as you can see on this scheme, what appears as a major limitation for the efficiency of this type of transport is the distance between the two membranes. And this is why it has been proposed earlier that this type of transport of lipid would be favored in specific areas of the cell where the membrane of two different compartments are close enough, are in close proximity, and in structures that were named membrane contact sites. So I will briefly describe what are membrane contact sites. So this is a scheme showing a typical textbook illustration of the different compartments of cells. So eukaryotic cells are an assembly of organelles, and they are often represented free-floating in the cytosol. Actually, it was already described in the 50s, but organelles are contacting physically each other, And this is shown here on this electron microscopy, where you can see, for instance, the endoplasmic reticulum that is pseudocolored in green, that is in contact, in physical contact, with mitochondria that are in brown, or with endosomes that are here in pink. And this is what how the organization of organelles is thought nowadays, with organelles that are involved in physical contact in membrane contact sites. And any pair of organelles has been described as making contact within this type of structure named membrane contact sites. So membrane contact sites are defined by the physical contact between two organelles with a low distance, that is most of the time below 30 nanometers. And these contacts are organized by proteins that are named tethers and that connect the membrane of two organelles. And they do that either by protein-protein interactions, so two proteins that are anchored in each of the each of the membrane of the two organelles that interact, or by protein-membrane interactions. And in the past decade, it has been shown that interorganelle contacts are involved in several cellular processes, such as organelle fission. It has been shown, for instance, to mediate the fission of mitochondria. They are involved in interorganelle communication, for instance, in the desactivation of EGFR in endosomes. And most of the mechanisms that have been described involve the transport of lipids and the transport of calcium. So there are many data that now show that membrane contact sites are a major contributor of lipid transport in cells. So I will now describe what the work we have done in our lab, and this work relates to a protein that is named STARD3. So STARD3 is one of these lipid transfer proteins that belongs to a family that is named the START protein family. So START proteins are 15 in human, from so STARD1 to STARD15, and 5 of them have been described as cholesterol binding proteins. And STARD3 is one of these 5 proteins. And STARD3 is composed of 2 domains, a star domain that binds cholesterol, and a mantle domain that is composed of 4 transmembrane helices that anchor the protein at the limiting surface of late endosome. And this leaves the star domain on the cytosolic side of late endosomes. So knowing that LTPs are, well, some LTPs act in membrane contact sites, we asked first whether STARD3 could be involved or could be localized in membrane contact sites. And to do that, we perform electron microscopy on cells, either control cells or cells that overexpress STARD3. And as shown here on these electron micrographs, what you can see is an endosome that with its characteristic ILVs on the inside of the endosome that makes contact with a tube of ER that is here. And in control cells, we see this kind of contact, but they are not so frequent. And most of the time, they are small at the surface of endosomes. In cells that overexpress STARD3, on the contrary, there is a huge recruitment of the endoplasmic reticulum at the surface of endosomes, as shown here on this electron micrograph. And there is a high increase in the number of contacts and in the surface of contact between endosomes and VR in these cells. So showing that STARD3 mediates the formation of ER endosome contacts. So we we then wanted to understand the mechanism of formation of these contacts. And we asked whether STAR-D3 possesses an interaction motif for ER proteins. And we found a potential motif at the end of the mental domain that correspond to a motif that is named FFAT, for 2-phenylamines in an acidic tract. And this motif has been described by Tim Levin as mediating interaction with proteins from the ER that are named VAP proteins. So VAP proteins are ER-bound proteins, so they are anchored in the limiting membrane of the ER by a transmembrane domain, and they possess a globular domain named MSP. And this MSP domain binds FFAT motifs, as shown here on this structure of the complex between the MSP domain in green and an FFAT motif in red. And the interaction between, and we showed that the interaction between the MSP domain of that protein and the FFAT motif of STAR-D3 mediates the formation of contact sites. And this is shown here on this scheme with STAR-D3 that is anchored at the limiting membrane of endosomes. It has an FFAT motif that interacts with VAP proteins, and the formation of this complex between STAR-D3 and VAP proteins allows the formation of contact sites between endosome and the endoplasmic reticulum. So knowing that STAR-D3 is found in contact sites, in ER endosome contact sites, we thought that maybe the star domain of the protein could mediate the transport of cholesterol between the two membranes, between the membranes of these two organelles. So to test this hypothesis, we wanted to detect free cholesterol in cells, and for that we used two different probes. So the first one is the GFPD4 probe. It's a protein probe that is derived from a toxin from bacteria that is named perfringolisine O. And this probe consists of the cholesterol binding domain of perfringolisine O that is fused to a GFP, so that it can be followed by fluorescence. And this probe recognizes regions with high cholesterol concentration. And this is shown here as an illustration. In control cells, it barely detects intracellulaire compartment, while in cells that are treated with a U drug, a drug that blocks NPC1, as TY mentioned, the inhibition of NPC1 induces the accumulation of sterol in endosomes. You can see that this probe is able to recognize cholesterol-laden endosomes in these cells. And the second probe that we used is Philippine, which is an antibiotic that is also fluorescent and that binds free cholesterol in membranes. And again, here you can see in control cells the labeling of the plasma membrane by Philippine, and in U drug treated cells, you can see the accumulation of cholesterol in endosomes that is labeled with Philippine. So we wanted to test the effect of the expression of STAR-D3 on the localization of cholesterol in cells. And so we expressed STAR-D3 in HeLa cells, and we labeled both late endosomes using LAMP1, which is here in red. We also labeled STAR-D3 in magenta, and we used the GFPD4 probe to label cholesterol. And we compared control cells to cells overexpressing STAR-D3. And as you can see on these higher magnifications on the right, in cells that overexpress STAR-D3, there is an accumulation of the GFPD4 probe staining in late endosomes that are both positive for LAMP1 and STAR-D3. We also tested the other probe, Philippine, and again, we labeled late endosomes with LAMP1 and STAR-D3 in magenta, and we labeled Philippine, which is here in blue. And as you can see again, with this other probe, we could detect an accumulation of cholesterol in LAMP1 positive and STAR-D3 positive late endosomes compared to control cells. Thus meaning that STAR-D3 promotes cholesterol accumulation in late endosomes. So together, this suggests that STAR-D3 is able to promote the accumulation of sterol in late endosome, and knowing that it is involved in the formation of contact sites between ER and endosomes, we wanted to know if these contact sites were essential for the transport of cholesterol and how it could work. And for that, we decided to reconstitute in vitro the machinery consisting of the STAR-D3 VAP complex, and to test if this machinery was autonomous for cholesterol transport. And this work was done in collaboration with Guillaume Drin in Nice, who perform in vitro lipid transfer assays using liposomes. And so the idea was to make artificial membranes that corresponds on one side to endosomes, so what we called endosome-like liposomes. So these liposomes, on these liposomes, we attach the start domain of STAR-D3 at the surface of the liposome. And we used a second population of liposome that we called ER-like. And here on these liposomes, we attach VAP proteins. So we have these two populations of liposomes, one that resembles endosomes, the other that resembles the endoplasmic reticulum. And in this assay, we used also fluorescent sterol, so DHE again, dihydroergosterol, and a threat acceptor in order to follow the transport of this sterol between the two populations of liposomes. And this transport was detected by threat. And by mixing the two populations of liposomes, we expected to reconstitute contact sites first, and then to detect the transport of sterol between the two populations of liposomes. So first, we tested if indeed in vitro we were able to reconstitute contact sites. And to do that, we performed dynamic light scattering experiment. So this experiment allows to detect the size of particles in solution. So at the beginning, we started with liposomes that were with a regular size of 100 nanometers, and we followed over time the possible aggregation of these liposomes that would be recapitulated by the formation of contact sites. So here in control liposomes, we could see that the size of the liposome at the beginning of the experiment in gray, or at the end of the experiment is blue, was the same. So all the liposomes had the size of 100 nanometers, all the particles. Then we used a mix of liposomes with a mutant form of that it was unable to bind with the start domain of STAR-D3. And again, we could not see any aggregation, so no contact site formation. While if we perform the experiment using the wild type form of that and the wild type form of STAR-D3, as you can see here, comparing the beginning of the experiment in gray and the end of the experiment in blue, and here over time, you can see that there is an increase of the size of the particle, showing that there is an aggregation of liposomes, meaning that STAR-D3 and VAP in vitro are able to recreate membrane contact sites. Then we tested the ability of these contact sites and the ability of STAR-D3 to mediate sterol transport in vitro. And for that, we used threat to follow the fate of DHE. So I remind you that DHE is fluorescent, and at the beginning of the experiments, it's only present in the ER-like liposome. And we followed the transport of DHE over time. so first in a system without protein, then using a soluble form of stardy-free protein, and you can see here this is in black. This system is quite inefficient to promote the transfer of sterol. And then using a second control, using a mutant form of VAP. In all these conditions, the transfer of sterol between the two populations of liposome is quite inefficient. And then we reconstituted contact sites. And now we had a huge increase in the speed of sterol transfer between the two populations of liposomes. And we could quantify that. And we end up with a speed of transfer of cholesterol that was at about 25 molecules of cholesterol that were transferred by protein per minute. Meaning that membrane contact sites between these two populations of liposomes enhance cholesterol transport, and that this complex between stardy-free and VAP is efficient and autonomous to transport cholesterol between ER-like membrane to endosome-like membranes. And so to summarize, what we showed here is that stardy-free builds ER endosome contacts, and it does that by interacting with ER resident proteins that are VAP-A and VAP-B. And within ER endosome contacts, stardy-free transport cholesterol from VR to endosomes. And this was the work of a former PhD student of the lab, Léa Villene. And with that, I will conclude, and I will thank all the members of the molecular and cellular biology of the breast cancer team and our collaborators, and in particular Guillaume Drin, IPMC Valbonne. And I will thank you for your attention and thank Philippe for his invitation.
Thank you very much, Fabien. Very nice. That's very, very interesting observation you have here. I think there's a question in the chat, so maybe either you or TY may answer that, or eventually Laurent if he wants to. Is the cholesterol content the same in all cell organelle membranes?
Yeah, so maybe I can come back to my scheme. So actually, no, it's not the same. So it's not the same, and it's highly enriched in some compartments, such as the plasma membrane. Here, so here it's really highly enriched in the plasma membrane. So it accounts for more than 40 mol% of total lipids in the plasma membrane. And in VR, VR is mostly devoid of cholesterol, so it's less than 3% of lipids in VR that are cholesterol. And in the different other organelles, it really depends. So mitochondria are not so much enriched in cholesterol, and lipid droplets are rich in cholesterol, but in esterified cholesterol.
Okay. Is there any other question in the chat or in the audience? I actually have another one. So you stain the cholesterol with Philippine or GFPD4, so that binds to free cholesterol, right?
Mm-hmm. Yeah.
So you're talking about movement of free cholesterol only. What about the esters?
Yeah. So yeah, with the probes that we use here, we could only detect free cholesterol. So regarding sterol esters, it's not so clear if they move inside cells. What is known is their final destination. It's known that they accumulate in lipid droplets. As TY mentioned, they are synthesized in VR or at the surface of lipid droplets by ECAT. And but once they are esterified, can they move out of lipid droplets, or do they need to be deesterified to move? This is not entirely clear.
Yeah, the question is about the filament. Does filament stain in cholesterol ester? My knowledge is no, it won't, because the cholesterol ester, I think the filament staining needs the 3-beta hydroxy group.
Yeah.
And once esterified, so the question is, filament does stain cholesterol. Oh, that's nice. What stains cholesterol ester? Stains cholesterol ester is like some kind of neutral lipid, like a Nile red. But then, how do you distinguish between cholesterol ester and triacylglycerol? I mean, is there any stain that can distinguish between cholesterol ester and not to my knowledge? That's just tough question. And the stain in the cholesterol ester, triacylglycerol, get into another technical problem, because there's some cholesterol ester gets stuck in the endolysosome. So is triacylglycerol, because the lipase may not completely hydrolyze cholesterol ester. And so some folks use the electron microscopy. I'm just looking into that. Presumably these neutral lipids get stuck in the endolysosome. They'll be membrane bound, and these lipid droplets, they should stay in the cytosol, and that's all I know. I wish there's a way to distinguish cholesterol ester and triacylglycerol. Not easy.
So there is there are some labs that use the fact that sterol esters are able to polarize light.
Yeah.
And using polarized light, they are able to distinguish sterol esters rich lipid droplets from triglyceride-rich lipid droplets.
I see.
It's really,
yeah.
There are only a few labs that are starting to do that.
Yeah, that would be very important. Yeah, to pin it down. Yeah.
There's other few questions in the chat. See, one of the three of you likes to answer this. Does the contact site were already shown to facilitate transfer of other content than lipids between organelles?
Yeah, so for instance, ions are known to be transferred between contact sites. So I can exemplify that with a complex between STIM1 and ORI1. STIM1 is a protein from the endoplasmic reticulum, and ORI1 is a calcium channel that is found in the plasma membrane. And these, when there is a depletion of calcium in cells, it induces a change in the conformation of STIM1 that allows its interaction with ORI1. And the complex between STIM1 and ORI1 occurs in contact sites between the ER and the plasma membrane. And in these contacts, calcium is transferred from the outside of the cell to the inside of the cell.
Okay. Another question, cholestérol is the only plasma membrane, in the plasma membrane comes from the ER only? Huh.
Yeah, this is a good question.
Huh.
Yeah.
Got the answer?
Yeah, this is a difficult question. So I don't know if TY has.
Well, you can address it, but you have this in vitro system. I like it a lot. I like the in vitro system. Eventually can answer that question. The question about cholesterol in the plasma membrane. I mean, the cholesterol in the plasma is actually heterogeneous. It's got this, it's got this lipid raft. Lipid raft is all over the place. This is Kai Siemens. I think the lipid raft really lives for a long time. Now people call it a lipid cluster, and it's heterogeneous. It's heterogeneous. So the question basically is, where does the lipid raft come from? That's I think that Kai Siemens has worked for long hours and cannot really address the question.
Yeah, I guess it's the best answer.
Don't have an answer. Don't have a good answer.
Yeah.
Yeah. In vitro cell culture will answer. In vitro system.
Yeah.
I like that.
I have another more general question. We heard of a functional difference between rodent and human liver, as well as monkey and human liver. Which monkey, to what aspect of metabolism could this be due to?
Yeah, this is very strange. Yeah, I guess the challenge came from my mouth. We found the rat, well, mouse and rat are very similar. For example, if one look at the mouse, the VODL, mouse, rat, monkey, they make a VODL. Look at the newly synthesized VODL. That's quite a bit of the cholesterol ester. And that cholesterol ester presumably either from ACAT1 or ACAT2. Now, in the monkey and the rat, it's mostly ACAT2. Now, how about the human? The human VODL is actually triacylglycerol rich. Their cholesterol ester is not very high. So, the newly synthesized VODL mostly is triacylglycerol, and then come out of the liver, and then where does the VODL pick up the cholesterol? It's the cholesterol ester exchange protein. Cholesterol ester exchange protein. Now the monkey may have, some monkey has a cholesterol ester exchange protein. Rat and the mouse doesn't have it. All right, so the cholesterol ester from the human, really it came from the exchange protein, mostly. It's not ACAT. And so the at that time, probably 20 years ago, if people want to reduce cholesterol, if you inhibit ACAT in the liver, it will block cholesterol ester. Well, it works all the time. It works all the time in the rat, monkey, and the mouse, rabbit, even rabbit, but it does not work in the human. So if you take a human clinical trial, cholesterol content in the blood did not reduce, so the company dropped it. So this one doesn't work. Well, it doesn't work because you did a bad experiment. You did not know that the metabolism between the human metabolism and the animal metabolism. Monkey looks quite similar to human, but the ACAT inhibitor reduces the cholesterol level in the monkey. It doesn't work in the human.
So, T.Y., I have a question actually for you, because so when you inhibit ACAT1, there is no ester that form. So the free cholesterol, the flow actually of the free cholesterol remain the same, right? So
well, that's a great question. It actually come down.
Oh, really?
Yeah, that's the key question. Depends on what system you deal with. Depends on your system, whether your cell system has efflux, does it have ABCA1, does it have caviar? Depends on the cell type that you look at. We look at the atherosclerotic plaque, read it with ACAT1, mild or ACAT1 knockout. And the total cholesterol content in the plaque actually reduces. That's a funny thing. If you do that, yeah, in the brain, the total cholesterol drop. So
but the same amount of esters enter the endocytic pathway, so you forms less. I mean, the lipase is activity is decreasing, or
somehow that cholesterol released. Okay, the cholesterol release from blocking cholesterol ester does some kind of a pathway. Get rid of cholesterol. The cell senses, aha, you're trying to build cholesterol to me. And so it increased the efflux, increased all these exocytosis, efflux. It stimulated ABCA1. Just get rid of it. And so that's a funny thing. We don't know. We don't have a pathway completely yet. That pathway needs to. It's a key question. Because you block it, the cholesterol. You think that by thermodynamics, what the hell the cholesterol is built up in the cell? It doesn't. It actually efflux out. So if you have a normal efflux system, there's no cholesterol buildup there. So there's no toxicity and stuff like that.
There's another question.
Yeah.
Thank you for these nice talks. So that's not a question, that's just a comment. I was more specifically wondering, that comes from the chat, I was more specifically wondering if endolysosome compartments could transfer some contents from endocytosed material, for example, to ER compartments through the contact site. Is it something that was already shown?
On the endocytosis material. Okay.
So it's not really clear. So what has been shown is, for instance, for cholesterol in some conditions, so we showed that there is a transfer of cholesterol from VER to endosomes. It has been shown in other cell types with other proteins, with other LTPs, that the other way around can happen. So meaning a transport of cholesterol from endosomes to VER, okay, mediated by, for instance, by ORC1M. But still, what has been shown so far is the transport of molecules, so molecule at a time, one molecule at a time from one compartment to the other. Then if this kind of structure could transfer, I guess that what Benjamin is asking here is more macromolecular complexes or material that is bigger than just one molecule of lipid. This so far has never been shown. But indeed, it cannot be excluded, but within contact sites, some bigger material could be exchanged, and maybe pieces of membrane, or I don't know. But this has so far never been shown.
I also have a question for Laurent. Laurent, are you still there? Laurent Calvier?
Yep, still there.
So I was just wondering whether the CR50 are actually decreasing the number of monocytes or just the adhesion of monocytes, because on the picture we had the feeling that there's less monocyte that are actually circulating.
Yeah, so I looked in the circulation. I don't think it affects the numbers in the circulation, but it definitely affects the number in the tissue. So either the subendothelial space during atherosclerosis or in the CNS during MS. So I clearly see that. I haven't checked that yet for arthritis, but I bet it's going to be the same. So it really, by decreasing the adhesion, it decreases also the number of leukocytes going into the tissue. So overall, the number of leukocytes in the tissue are less. Yeah.
There's another question for you, Laurent.
Yep.
In your opinion, how do PCSK9 inhibitors affect the described mechanism in atherosclerosis, knowing that PCSK9 inhibition increases ApoE2 endothelial cell surface expression?
Yeah, that's a good question, because that's true. There is interaction between PCSK9 and ApoE2. So that's something I haven't checked yet. Like if you decrease the expression of directly the receptor instead of decreasing the expression of the ligand trilin, if it does the same. That it's a bit tricky, because actually there is different receptors for renin. Like the VLDL-R is also a receptor for renin. Mainly, it looks like in endothelial cells that doesn't matter so much, but maybe in other tissues that might have an effect. So it's really difficult to answer this question, and I haven't looked into it yet.
Okay. Okay. Is there any other question from I don't see any other on the chat. Maybe somebody wants to ask another question. Otherwise, thank you very much, TY. Thank you, Laurent.
Thank you.
Thank you, Fabien. That was really, really interesting talks, beautiful works, beautiful data. And thank you for all the participants for being here tonight and sharing with us all this beautiful data. I wish you a good night, good evening, and see you next webinar of the NSFA. Bye-bye.